is a clinician but also a researcher at the University of Sydney and the Kolling Institute. We're now working with Carol on our lead candidate 8114 and looking at it for potential treatment in kidney fibrosis. But Carol's going to give us an overview of what ion channels are. She's got significant experience in that space working as a kidney clinician. Thank you, Carol.
Thanks, Sam. Yeah, so I just might add a bit about myself. I guess I did my nephrology training and then I did a PhD. And in my PhD, I looked at inhibiting the sodium glucose linked transporters and demonstrated that they were protective in the kidney and lowered the blood glucose at the same time. I published that, I moved on, and 20 years later, every company has an SGLT2 inhibitor, and I didn't patent my findings, and I recognised I had a serious deficiency in my approach to science. Well, now I'm on the advisory boards of Merck and J&J and Boehringer Ingelheim and AstraZeneca, etc., because all of them have SGLT2 inhibitors, and I had done the physiology at the time and then subsequently the molecular physiology of SGLT2 inhibitors. So I sort of realised I needed to take a different tack and bring together my clinical work and my scientific work so that I could actually see what was actually going to help patients into the future, and then try and develop I guess, an interest in helping pharma, biotech, etc., to bring those to market for patient benefit. So I'm not particularly interested in starting a company for myself or whatever, but I am interested in helping biotech and pharma to develop new therapies. So I guess, my own laboratory has been interested in, supporting, for instance, patent positions of small biotech. And we've done some work, for instance, with Pharmaxis to bring their amine oxidase inhibitor to market, and was purchased by Boehringer Ingelheim for a fair amount of money. I've worked with Darren Kelly when he brought his FT11 to market, purchased by Shire, etc., now reacquired by Darren. Anyway, so I have a particular interest in AdAlta because I think they have some fabulous technologies that are really going to help my patients who have kidney disease. So what I'm interested in at the moment is ion channels, and ion channels are basically, I'll have a bit of a discussion about them, but they're critical for pretty much every biological process. The people would recognise ion channels as calcium channel blockers, for instance, to help in blood pressure and things that modify cardiac arrhythmias, for instance, different muscle disorders, cognition, etc. So basically ion channels allow ions to come from the outside of the membrane to the inside of the membrane, or vice versa. And then through that, they actually then, particularly calcium inside the cell or inside the subcellular area, is then the signal to start up a whole lot of cytokine delivery, really inflammatory processes that underpin kidney disease. They've been quite difficult to translate, and obviously there's a lot of similarity in various ion channels. So that if you target an ion channel with a small molecule, you're often likely to have off-target effects. So the spectacular problem was with Posicor, which was a fabulous blood pressure lowering agent, but it actually caused sudden cardiac death. So there was a huge investment. I think it was BMS at the time, and everybody died. So blood pressure was fabulous, but the patient died. What?
Except BMS.
Yeah. And I remember when antibodies were first generated, they had a huge amount of off-target cytokine storms, which have still been the case. So you do need to get these very specific antibodies that don't cause an antibody reaction to the antibody, so they have to be tiny, and you can't have off-target effects with them because it can be, spectacularly problematic for the patient and for the company. So ion channels can be difficult to target. They can also be very easy to target. If they're easy to target, nothing's easy, so there's a problem. And this similarity between the ion channels means that often ion channels are thought of as families rather than as the channel, and the family has broad effects in the brain, the heart, etc. So you want an ion channel that can be specifically targeted to the specific organ that you want, and not have off-target effects, which is why AdAlta's technology is great, because it can target a channel without targeting a family of channels, because you don't want that to occur. Now, my interest, you raised fibrosis, is kidney fibrosis, because fibrosis in the kidney is the end result of any disease that affects the kidney. So no matter what your disease is, it turns out with all this, green stuff, which is just scarring in the kidney. So you want to try and stop the scarring, and it's irrelevant what disease you're looking at. So it could be a very rare kidney disease, or it could be diabetes. Now, my interest is in diabetes, because that's where the majority of people who have kidney disease, if you look at it population base, about 35% of people on dialysis will have it because of diabetes, and about 16% will have it because of high blood pressure. Both of those should be completely preventable forms of kidney disease. And if you look at the Medicaid cost in the US, kidney disease accounts for the greatest cost to the Medicaid system. That's partly because they've chosen to support kidney disease in America, but the costs are increasing. Now, most people with kidney disease don't end up on dialysis, they die before that. So only a small proportion of people with kidney disease end up on dialysis, but most people get cardiac disease. And cardiac disease people think of as being caused by high blood pressure and by diabetes, but in fact kidney disease is the strongest predictor of whether or not you're going to get cardiovascular disease. So this is what's happening to dialysis. And if you think of it, a small proportion of people with kidney disease end up on dialysis. You look at the causes. In general, the non-diabetic causes are falling and the diabetic causes are increasing, and it's largely because of type 2 diabetes, and that's where we work. So if you look at the prevalence of diabetes, basically in 1990, they assumed that there was going to be about 333 million people with diabetes in 2015, and that number had increased to 415 billion people. So that the estimates of just, are twice as high as what people thought was going to be the case, and that's reflected in the number of people with kidney disease as well. So if you look at what our current treatments are, it's basically been really blockade of the renin-angiotensin system with ACE inhibitors or angiotensin receptor blockers. And that was demonstrated really in the 1990s, where in people with type 1 diabetes there was a 28% reduction in the development of end-stage kidney disease. Up until the SGLT2 inhibitors, there's really been nothing, and the EMPA-REG study with the secondary analysis came out as showing that that was going to be beneficial in kidney disease. Canvas then subsequently demonstrated the same thing, so that was with empagliflozin and then with canagliflozin, but they're both secondary endpoints. So now there's the Credence study that will probably report in end of next year, I think. And I'm on that steering committee internationally, but there's really been nothing else for the last 25, 30 years that are going to be helpful for people with kidney disease. And although there's a 28% risk reduction, there's a large residual risk. So we've been interested in looking at potassium channels, and the first one we looked at was this voltage-independent potassium channel that's activated by intracellular calcium. So it's the KCA 3.1. It's various other names, KCN4, the Gardos potassium channel. But we've been interested in it because if you get this calcium influx, then that is what triggers a whole heap of inflammatory processes. So we and this really is just looking at you basically get the potassium exits the cell and calcium comes into the cell and then fires off all of these various intracellular processes. So a few years ago we did some work initially looking at a knockout mouse model. So the KCA 3.1 was knocked out. There's no phenotypic abnormality in the mouse at all. And we looked at TRAM34. Now TRAM34 is not a particularly good blocker of the calcium, the potassium activated calcium channel, but it was useful as a tool. And basically what we demonstrated was that this channel was very overactive, so the brown dots are the channel. So this is the normal, this is in a diabetic animal, so that there's an increase in this particular channel here. And if you use this drug, then you can decrease that channel activity. And by virtue of decreasing that channel activity, we demonstrated that it could markedly inhibit, remember the green bits, the scarred bits, so that comes back completely to normal. And then this is collagen, which is an extracellular matrix protein that goes to make up a lot of this fibrotic material. So all of this here has now been, it's not present if you knock out that channel. Well, we're interested in this channel here, the KB1.3, because if you look at that, this is one that's particularly on the mitochondrial membranes, and as I say, the mitochondrial membranes is where cellular bioenergetics come into it. So you're looking at inhibiting oxidative stress, which is something that we know promotes kidney disease, promotes a lot of disease. And all of these things here, well, not all of them, but a lot of them are due to this signalling through the mitochondrial membrane that traditional small molecules can't target and large antibodies can't get to. So you can see this here is fabulous for metabolic disorders, diabetes or pre-diabetes, etc., obesity, but also it has shown to slow the development of kidney fibrosis because of this issue with potassium and calcium signalling. So we're quite interested in that. We just have to get AdAlta to be interested in targeting ion channels. Sorry?
Targeting KB1.3, what off-target effects would you be concerned about?
Well, I think that a lot of the peptides and other things have been shown to be problematic because of off-target effects. But you look at what mutations have been shown. There can be abnormalities in blood disorders. You can get problems with cell swelling. And occasionally, there's a theoretical suggestion that you could get high blood pressure because you can get vascular constriction, but animal models haven't demonstrated that you get high blood pressure. So I'm not thinking that there's going to be particular problems. There's no known cardiac conduction defects with it, which would be a theoretical problem with all of the ion channels. And there's no abnormalities neurologically that you would be thinking of, but they're always going to be the problems. When we, as I say, in the knockout models, there doesn't seem to be any problems, and they do seem to have a resistance to obesity. So if you high fat feed them, they don't tend to become obese. So sort of interesting. Anyway, there's a potential for it to be involved in a whole lot of different disorders, not just in kidney fibrosis, which is my main interest. So if you look at, the lymphocyte type diseases, and transplantation is something that obviously we get involved in quite a lot. And as I say, I remember the anti-CD3 antibodies where you remember people would almost die, but then their transplant would function, and then you could only give it to them once because they'd get an antibody against the antibody. But we use a lot of cyclosporin and tacrolimus to target the calcineurin pathways and the nuclear activation. But this would be another way to target that pathway that would be more proximal. So I think it's certainly of interest. Just to give a little bit of an update on what we've been doing to date with the CXCR4 antibody, we've also demonstrated that that's particularly upregulated, as are a lot of the G protein coupled receptors in kidney disease. And we demonstrated that the AdAlta lead candidate binds to it only binds to tissue when it's upregulated, so it binds quite well in the kidney tissue. And then we looked at extracellular matrix, and so these are two of the major extracellular matrix protein, fibronectin and collagen, and we demonstrated just in dose response studies that we can show an upregulation if we add transforming growth factor beta. Transforming growth factor beta underpins the majority of fibrotic processes in all organs really, but in the kidney in particular, and this is just done in cell culture, so we add transforming growth factor beta to our proximal tubular cells. The proximal tubular cells make up 90% of the kidney, and so you get an increase in the matrix production from these particular cells that is blocked by co-incubation with the AdAlta product. And basically, in summary, we've shown a reduction in the fibrotic markers, and then this year we were given $768,000 from NH and MRC to proceed with these studies with AdAlta. And that's actually quite amazing that NH and MRC would support these studies with a company, because often, you go to these panels and somebody will say, well, why isn't the company paying for, these studies? So it obviously excited NH and MRC, and we're pleased to be able to progress with it. When you look at all the things that are done in kidney disease, a lot of them fail because there's so many processes that can actually lead to that end organ fibrotic study. So when we look at the processes underpinning kidney disease, they tend to be oxidative stress, metabolic processes, and hemodynamic processes. So if you look at anything, you have to really look at it on the background of existing best practice therapy, which is inhibition of the renin-angiotensin system. The metabolic diseases, you can prevent them, we think, with the these ion channels and the some aspects of oxidative stress, but also there's inflammatory aspects through inhibiting this. So it's always going to be a combination strategy here, because otherwise if you block this, then this is still going on. And you can see all of these processes. So we think that we might need a combination of therapies, and something like a ion channel strategy with a blockade of the inflammatory response as well, we think will be effective. So basically that's the summary. So, so I
Can I
finished.
just ask, when you're talking about targeting things that are sort of on the endoplasm in particular?
The profile of ion channels and GPCRs that change from normal airway epithelium to epithelial mesenchymal transition is phenomenal. And I think you've got a nice example there with CXCR4. But have you looked at the whole stack of trip channels, whether they change?
No, I haven't looked at any of
That'd be
them.
worth doing. And you could target that with AdAlta approaches.
I think Mick will talk about it in a minute, John.
I don't know whether it's necessarily that one, but there are other ones.
Yeah.
No, I haven't. I mean, originally I did a whole lot of work in using electrophysiology and BCECF, looking at changes in intracellular pH, etc. But I haven't looked at it
Well,
with
I mean,
any of
after where
these.
things have changed, basically everybody thought ion channels was nerves, conductors, that type of thing. But actually ion channels are very important in non-voltage activated systems. And they really do play a key role.
No, agree
To your
entirely.
point, that's why 40% we don't know the ligands, right? Because only the CNS people care about them historically.
Yeah, but you can use systems to activate them without knowing the ligand and things like that. And I mean, the biggest advances, other than antibody technology, is the ability to do high throughput screening of ion channels. In other words, the big problem used to be electrophysiology would take forever to do. But now you can do multiple thousands with the systems available now.
Carol, do you have access to to small molecule pharmacology approaches? I mean, so you can do knockout work, right? That's reasonably straightforward. But have you got access to small molecule libraries, or trying to identify
With partners.
specific agonists or antagonists
Yeah.
these channels to pharmacologically modulate them?
Yeah, there's been several big pharmas that have had interests in ion channels, like AstraZeneca, I guess, has been one of the people that have been most interested. But their technologies weren't able to access the subcellular membranes. So often they've fallen over, but there's a guy who is one of the chief scientists at Astra that's sort of looking at some of this work. And I think, you know, the nanobodies that Bianca spoke about there's some interest in looking at the nanobodies with these channels as well. So the answer is yes, but I'd have to get a partner to do it. I don't have the libraries myself.
But you could do it the other way. You just do a global GPCR or global ion channel screen using quantitative PCR on your isolated cells and show what's there.
Yeah.
And then come in with your compounds around it.
Yeah, that's right.
That's what we've done on the airways.
Yeah, and people are interested because we culture human cells. So people that have nephrectomies for whatever reason, we get the rest of the kidney that would otherwise end up in the bin.
Yeah.
And it's sort of interesting because in the US, you know, there's scientists and there's clinicians, and almost never the twain shall meet. And so people are, you know, they're always amazed. Why would you bother doing a PhD if you're a doctor? And you know, like it's sort of unusual, and they're done in different buildings, whereas, you know, we're all in the same building.
Yeah,
So
it's really important when you
yeah.
put it together.
So, you know, we do use some, you know, perpetuated, immortalised cells, but they usually have problems with them, you know, in that they translocate their ion channels from apical to basolateral.
Yeah, they transfer yourself to those type of cells.
Yeah. Well, that's
The
right. You have to use a
obvious is
simian
the cells
virus or
you're
something.
working with.
Sorry?
Primary culture,
Yeah.
that's the key. Yeah.
And we've well characterised them, you know, because sometimes, for instance, if you're looking at toxins, the organic anion transporters aren't on the right side of the cell, so you can't actually look at, you know, paraquat poisoning or whatever you want to be looking at in immortalised cells. It doesn't work.
Carol, sorry, your NHMRC grant, congrats. That for your grant, will we see phase 1 data? I mean, what does it allow you? What does it fund you for?
Yeah, it's basically animal work. It's not going into phase 1 data, but that would be, you know, that would be fabulous if we could do that. But it really is to, you know, I'm sure that the patent position of the CXCR4 antibody will improve if our studies look as though they're positive. I guess that's why I'm here. Yeah.
Could you just summarise why you think a combination therapy would be more likely?
Because I think if all of the drugs that have, you know, there's a huge number of drugs that have fallen over in kidney disease. You know, the anti-TGF-beta antibodies, the you know, CTGF antibodies, small molecules against X, Y, and Z. Because you target one part of the pathway and another part will come in. So there's a lot of redundancy in the mechanisms towards fibrosis. So I think you do have to target the hemodynamic, the metabolic, and the oxidative stress all at once.
It takes back in time. You're working on SGLTs a long time ago, and suddenly GSK and others prick up their ears and start paying attention. What was the catalyst for them?
Catalyst for who?
For the big pharma guys to pick up on the SGLT?
Well, I think they picked up on it because it lowers blood glucose and it's independent of insulin. So all of the other therapies, well, not all of them, but the majority of them work by either increasing your endogenous insulin production or increasing your insulin sensitivity, whereas SGLT2 inhibitors have a completely different mechanism of action in that you're basically stopping the reabsorption of glucose from the kidneys back into your bloodstream. So it doesn't matter about insulin. You can have diabetes for 40 years, your pancreas can be completely destroyed and they'll still work. So they started off thinking that you couldn't have it, or they wouldn't be effective if your kidneys didn't work very well. And I guess, you know, in the EMPA-REG study, which was the cardiovascular outcomes study, because you'll recall there's been some issues about some diabetic drugs perhaps increasing cardiovascular risk. So now all of the studies have to have a cardiovascular safety profile. And as a consequence, they demonstrated that there was some benefit in kidney function. And in fact, there's about a 40% reduction in the development of end-stage kidney disease with SGLT2 inhibitors in the secondary analyses, so they weren't targeting that. So the Credence study now looks at people who have down to about 30% of kidney function, and it would seemingly be, because the EMPA-REG study and the Canvas studies, including Canvas-R, which was a renal study, have both very similar degrees of renoprotection. So it then led to, well, maybe they have mechanisms of benefit other than the lowering of glucose. So there's a whole heap of, you know, theories why that might be the case. But the pharma companies got initially interested because of the glucose lowering effects, and the diabetes market is this big. So the kidney benefits are sort of a happy side effect of that. And then because I'd done all that work about it, I became the world expert on SGLT2 inhibition and renal protection.
Was there a moment when the scales fell from their eyes and they realised they needed to pay attention to Carol back in Sydney?
Yeah, well, that was amazing, because all of a sudden I started to, you know, people were interested in asking me to give talks at various meetings and whatnot, and I had to convert my slides into PowerPoint. So, I mean, how many times do you get out your PhD slides and then have to think, I need to do something with this here? So,
Didn't take the carousel with you.
That's right. I was taking a photo of my slides, yeah. So, yeah, so it was a big lag phase, I think, as Bianca suggested. You know, like it was a long while before there was any interest.
Just a couple of weeks ago, one of the analysts in the Citigroup, I remember correctly, they wrote a big report on the SGLT2 and saying how the market is underestimating this class, particularly for the kidney, and how this is going to be so much bigger for AstraZeneca. And to your point, we always looked at that class as an interesting one, particularly for China and diabetes. I think the companies like it because it's relatively safe. It also, I think, causes some weight loss.
Yeah.
And so it's this perfect diabetes.
And lowers the blood pressure because you lose sodium
So
with it.
it's this kind of great thing, and now I think there was a deep dive analyst report on why this class should be so much bigger, and so I think that is now kind of perpetuating itself to this great new solve that everything is going, so hopefully remain safe. So
Yeah, so now it's being used in heart failure, for instance, and there's trials in, you know, obesity independent of glucose lowering, and kidney disease independent of diabetes, and, you know, so it's sort of almost like, you know, it's not going to be beneficial for all of those things, but certainly there's trials going on. And the EMPA-REG study and the Canvas study were both done in people with high cardiovascular risk. So the DECLARE study, which is what the Astra study is a much bigger study, but in people with low cardiovascular risk. So it will be able to tell whether or not there are these other benefits there. I think there's 17,000 people in the DECLARE study.